The human heart has over 5 billion cells, and when someone suffers from heart failure, the tissue has very limited regenerative ability. And so computational modeling basically helps us to predict outcomes that would allow us to grow these cells optimally while reducing the time and expense in the lab. One important factor here is glucose, as that acts as an energy source for the cells to grow. The consumption of this glucose leads to the production of lactate. And when lactate levels are high, they lead to toxicity, which will lead to cell death. So these cells are grown in a dynamic system, which is a bioreactor. In the bioreactor, there's an impeller which generates shear stress. Now, moderate levels of shear stress have been shown to help enhance growth of the cells. However, when the levels of shear stress are high, they will disrupt the cell membranes and lead to cell death. A high energy dissipation rate will lead to high shear, which will lead to cell death. On the other hand, a low energy dissipation rate will mean that there is not enough cell mixing, and so there's no uniform distribution of nutrients. By capturing all these biological and mechanical factors in the model, we can pinpoint the optimal and most cost-effective conditions for growing cells. And so this means that researchers can design better and more affordable experiments, moving us closer to creating reliable cell-based therapies for repairing heart tissue.